Hi viewers, welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding the translation mechanism from the molecular basis of inheritance in detail. So here what is translation and what exactly happens during translation? After the successful synthesis of mRNA during the transcription mechanism, the specific mRNA it is ready to participate in case of the translation in order to synthesize one specific polypeptide chain or protein. And here during the translation mechanism, polymerization of amino acids takes place in order to synthesize one single polypeptide chain or protein. Is it clear? So let me write it on the board. After the synthesis of mRNA during transcription, the specific mRNA is ready to participate in translation in order to synthesize single polypeptide chain or protein. So here during the translation mechanism, polymerization of amino acids takes place. So let me write it here. During translation, polymerization of amino acids take place in order to synthesize polypeptide chain or protein. Is it clear? So here in between the two adjacent amino acids, the specific bond which is present here, it is nothing but what? Peptide bond. Is it clear? So let me write it here. Peptide bond is present in between two adjacent amino acids. And here the formation of the peptide bond in between the two amino acids, it is an energy dependent process. Is it clear? Formation of peptide bond is energy dependent process. Is it clear? So here in order to organize the translation mechanism, both in case of prokaryotes and even in case of eukaryotes, there is a requirement of certain raw materials here. So here what are the various raw materials which are required for organizing the translation both in case of prokaryotes and even in case of eukaryotes? Here the various raw materials which are required, these include mRNA, tRNA, ribosomes, and the other one is amino acid tRNA synthetase. Is it clear? So let me write it here. The various raw materials which are required for organizing the translation, these include mRNA, the other one is tRNA, and the other one is ribosomes, and the last one is amino acid tRNA synthetase. So here all these are the various raw materials which are required for organizing the translation mechanism or the translation process both in case of prokaryotes and even in case of eukaryotes. Is it clear? So at first, if we consider the mRNA, so here what is mRNA or how we can define the specific mRNA? mRNA, it is a single stranded RNA which is most commonly synthesized during the transcription mechanism by taking the advantage of a specific enzyme which is nothing but DNA dependent RNA polymerase. Is it clear? And here the specific mRNA which successfully synthesizes during the transcription mechanism by taking the advantage of RNA polymerase, it is a single stranded RNA. And here if the specific mRNA, if it is double stranded, under such condition, from one particular gene, two polypeptide chains or two proteins are synthesized, which is quite against to the central dogma of molecular biology. So the reason why the specific mRNA which successfully synthesizes during the transcription mechanism, it is a single stranded RNA. And in addition to this, if the specific mRNA, if it is double stranded, under such condition, the specific subcellular factory, which is nothing but ribosomes, in order to synthesize the specific polypeptide chain, it is unable to recruit to the mRNA if the specific mRNA is double stranded. Is it clear? So let me show it here. So let us assume it is the mRNA here. Is it clear? So here if the specific mRNA, if it is single stranded, only under such condition, the specific subcellular factory which is responsible for organizing the translation mechanism, which is nothing but ribosomes, it is capable to recruit to the mRNA. And here if the specific mRNA, if it is double stranded, under such condition, the specific ribosomal subunit, it is unable to recruit to the mRNA. Is it clear? So let me show it here. So here these are the two ribosomal subunits which successfully recruit to the mRNA in order to organize the translation mechanism or the translation process. And here if we consider the specific mRNA, so here mRNA, likewise the other nucleic acids like DNA and tRNA, even the specific mRNA, it is also a type of nucleic acid. And basically the particular mRNA, it contains many number of nucleotides like AUGC. Is it clear? So let me write it here. Basically, mRNA contains various nucleotides like AUGC. And here all the nucleotides in case of mRNA, they are not expressed in the means of singlet form or doublet form, but here they are expressed in the means of a triplet form, which is termed as codon. Is it clear? All the nucleotides in mRNA are expressed in the means of triplet form, which is nothing but codon. Is it clear? So here many number of codons, they remain very adjacent to each other in case of one particular mRNA. So here each and every codon in case of the mRNA, they are very specific to one particular amino acid which do exist in nature. Is it clear? So let me show it here. Let us assume there is one specific codon which is nothing but AUG. Is it clear? So here to the specific codon known as AUG, one specific amino acid known as methionine, it is very specific. Is it clear? All the codons which are present in case of the mRNA, they are very specific to one particular amino acid which do exist in nature. And here all the codons which are present in case of the mRNA, they are capable to encode one specific amino acid by taking the advantage of a specific molecule which is nothing but what? tRNA. Is it clear? So here by taking the advantage of a specific adapter molecule known as tRNA, each and every codon in case of the mRNA, they are capable to recognize one specific amino acid which do exist in nature. And here the specific mRNA, it not only contains the sense codons, and in addition to the sense codons, even the specific mRNA, it also contains certain stop codons like UAG, UGA and UAA. Is it clear? So let me write it here. mRNA in addition to the sense codons, it also contains stop codons or termination codons like UAG. So let me write it here. UAG, UGA and UAA. And in addition to the sense codons and the stop codons, even the particular mRNA, it also contains additional sequence which is present both at the extreme 5' end and as well as the extreme 3' end. And here it is termed as what? 5' untranslated region and the other one is 3' untranslated region. Is it clear? So let me write it here. mRNA also contains 5' UTR. So here UTR stands for what? Untranslated region. Is it clear? And it also contains 3' UTR. So here most commonly the specific 5' UTR, it is present prior to the start codon or the initiation codon, and whereas 3' UTR, it is present after the stop codons or the termination codons. Is it clear? So here this is regarding what? mRNA. And the next specific raw material which is responsible for organizing the translation mechanism, here it is nothing but what? tRNA. So here what is tRNA? So here tRNA, it is the specific RNA which is most commonly synthesized during the transcription mechanism by taking the advantage of a specific enzyme which is nothing but what? Bacterial RNA polymerase in case of prokaryotes, but whereas RNA polymerase 3 in case of eukaryotes. So here by taking the advantage of these specific enzymes, the specific adapter molecule which is nothing but tRNA, it successfully synthesizes during the transcription mechanism both in case of prokaryotes and even in case of eukaryotes. Is it clear? So let me arrange the things here. And here the next specific raw material which is responsible for organizing the translation, here it is nothing but what? tRNA. So here what is tRNA here? tRNA, it is the specific RNA which most commonly synthesized during the transcription mechanism by taking the advantage of bacterial RNA polymerase in case of prokaryotes and whereas RNA polymerase 3 in case of eukaryotes. Is it clear? So let me write it here. tRNA or transfer RNA. It synthesizes during transcription mechanism by taking the advantage of bacterial RNA polymerase and RNA polymerase 3 in case of eukaryotes. Bacterial RNA polymerase and RNA polymerase 3 in case of eukaryotes. So here what is the role of tRNA here? The specific tRNA, it acts as an adapter in between the codon of the mRNA and as well as the amino acid. Is it clear? So let me show it here. So here let us assume it is the mRNA. So here as I have already mentioned you, the specific mRNA, basically it contains many number of codons. So here each and every codon in case of the mRNA, they are very specific to one particular amino acid. But here each and every amino acid itself, it is unable to recognize the codon in case of the mRNA. So here the amino acid, in order to recognize the codon in case of the mRNA, it takes the advantage of a specific adapter RNA molecule which is nothing but what? tRNA. Is it clear? So let me show it here. Let us assume some of the codons like AUG, GUG, UUG and CUC. So here each and every codon in case of the mRNA, it is very specific to one particular amino acid. But here the amino acid itself, it is unable to recognize the codon in case of the mRNA. Amino acid, in order to recognize the codon in case of the mRNA, it takes the advantage of a specific adapter RNA molecule, which is nothing but what? tRNA. So here it is nothing but what? tRNA, right? So each and every tRNA, they are very specific to one particular codon in case of the mRNA. And here, by taking the advantage of tRNA, each and every amino acid, it is capable to recognize the codon in case of the mRNA. So here for the very first time, this particular hypothesis, it was postulated by Francis Crick. Francis Crick, for the very first time, he postulated that each and every amino acid themselves, they are unable to recognize the codon in case of the mRNA. Amino acids, in order to recognize the codon in case of the mRNA, they take the advantage of a specific adapter RNA molecule, which is nothing but what? tRNA. And here the particular hypothesis, here it was termed as what? Adapter hypothesis, which was first explained by Francis Crick. Is it clear? So let me write it here. Francis Crick for the very first time postulated that each and every amino acid themselves are unable to recognize the codon in mRNA. Recognize the codon in mRNA. And here the specific amino acids. So let me write it here. Specific amino acids, in order to recognize the codon in mRNA, takes the advantage of adapter RNA or tRNA. And here the specific hypothesis, here it was termed as what? Adapter hypothesis. Is it clear? And basically the particular tRNA, which is responsible for encoding the codon in case of the mRNA, it has two specific sets, and these include, one is the acceptor stem and the other one is anticodon loop. Is it clear? So let me write it here. Basically, tRNA has two specific sites for organizing translation. And these include acceptor stem, and the other one is anticodon loop. Is it clear? So here each and every amino acid, they are capable to attach to one particular tRNA exactly at the acceptor stem. Then in the same way, anticodon loop. So here it is nothing but what? Anticodon, right? So each and every tRNA, they are capable to recognize the codon in case of the mRNA by taking the advantage of a trinucleotide sequence, which is nothing but what? Anticodon. So here both the acceptor stem and as well as the anticodon loop, they play an important role in organizing the translation mechanism, both in case of prokaryotes and even in case of eukaryotes. And here the specific tRNAs which most commonly participate during the translation mechanism, basically they are of two different types, and these include, one is the initiated tRNA and the other one is the amino acid tRNA. Is it clear? So let me write it here. tRNA which generally participate during translation are of two different types. And these include, one is the initiated tRNA. And the other one is amino acid tRNA. So here, why it is termed as initiated tRNA here? Because this is a specific tRNA which is responsible for initiating the translation mechanism both in case of prokaryotes and even in case of eukaryotes. Is it clear? The specific tRNA which is responsible for initiating the translation mechanism, here it is termed as what? Initiated tRNA. And here the specific tRNA which is responsible for the formation of peptide bond in between the initiated tRNA encoding amino acid, here it is termed as what? Amino acid tRNA. Initiated tRNA, it is responsible for initiating the translation mechanism, and whereas amino acid tRNA, it is the specific tRNA which is responsible for the formation of peptide bond in between the initiated tRNA encoding amino acid and as well as the amino acid tRNA encoding amino acid. Is it clear? So here this is regarding what? tRNA. And the next specific raw material which is responsible for organizing the translation mechanism, here it is nothing but what? Ribosomes. So here what are ribosomes here? Ribosomes are ribonucleoproteins. Chemically they are composed of RNA and as well as proteins. Is it clear? So let me arrange the things here. And here the next specific raw material which is responsible for organizing the translation, here it is nothing but what? Ribosomes. So here what are ribosomes here? Ribosomes are ribonucleoproteins. Is it clear? So let me write it here. Ribosomes. Ribosomes are ribonucleoproteins. Ribosomes are ribonucleoproteins. They are chemically composed of RNA. They are chemically composed of RNA, and as well as 80 different proteins. And 80 different proteins in prokaryotes. 80 different proteins in prokaryotes. So here the specific ribosomes which generally do exist, basically they are of two different types, and these include prokaryotic ribosomes and the other one is eukaryotic ribosomes. Is it clear? Ribosomes, they can be prokaryotic ribosomes and even they can be eukaryotic ribosomes. So here the prokaryotic ribosomes, they are 70S ribosomes. Is it clear? Prokaryotic ribosomes are 70S ribosomes. 70S ribosomes. And here the 70S ribosomes, basically they contain two specific subunits, and these include 50S and the other one is 30S. 50S is the largest subunit, and whereas 30S is the smallest subunit. And in the same way, in case of eukaryotes, the specific ribosomes which generally do exist, they are termed as what? 80S ribosomes. So let me write it here. 80S ribosomes. And here the 80S ribosomes, likewise the prokaryotic 70S ribosomes, they also contain two specific subunits, and these include 60S and the other one is 40S. So let me write it here. 60S, which is the largest subunit. largest subunit, and the other one is 40s, which is the smallest subunit. So here S stands for what? S stands for Swedberg unit, which has obtained during the sedimentation coefficient of the centrifugation mechanism or the centrifugation process. And here, basically the particular ribosomes, it has three specific sites, and here these include A site, P site, and the other one is E site. Is it clear? So let me write it here. Basically, the ribosomal subunits has three specific sites. And these include A site, P site, and the other one is E site. So here A site, it is the specific site which is responsible for the binding of amino acid tRNA. And here P site, it is the specific site, or it acts as a hotspot for binding of peptidyl tRNA. Is it clear? Then in the same way, E stands for what? Exit. So here these are the three specific sites which are most commonly present in case of the ribosomal subunits. And here the specific ribosomal subunits, they remain in dissociated form when the specific ribosomal subunits, when they won't participate in case of the translation mechanism. Is it clear? The ribosomal subunits, both in case of prokaryotes and even in case of eukaryotes, they remain in associated form only under the active conformation. When the ribosomal subunits, when they participate in case of the translation mechanism, only under such condition, the ribosomal subunits, they remain in associated form. But here, once when the ribosomal subunits, once when they won't participate in case of the translation mechanism, they remain in dissociated form. Is it clear? So let me write it on the board. Ribosomal subunits remain in associated form during active conformation, and they remain in dissociated form during inactive conformation. Right? And here, basically the specific ribosomal subunits, they contain specific ribozymes which are responsible for organizing the peptide bond. And here these include 23S rRNA in case of prokaryotes, and whereas 28S rRNA in case of eukaryotes. Is it clear? So let me write it here. Ribosomal subunits has ribozymes for organizing peptide bond. And here these ribozymes, they are nothing but what? 23S rRNA in case of prokaryotes, and whereas 28S rRNA in case of eukaryotes. So let me write it here. 23S rRNA in case of prokaryotes, and whereas 28S rRNA in case of eukaryotes. So here these specific RNAs, they are termed as ribozymes. So here why they are termed as ribozymes here? Because basically these are the RNAs which are having the enzyme activity or catalytic activity. Is it clear? Ribose nucleic acid which is having the enzyme activity or catalytic activity, here it is termed as what? Ribozymes. Is it clear? So this is regarding what? Ribosomes. And the next specific raw material which is responsible for organizing the translation, here it is nothing but what? Amino acid tRNA synthetase. So here what is the role of amino acid tRNA synthetase? We will be studying regarding the mechanism of translation. So here while explaining the mechanism of translation, surely I will explain you what is the role of amino acid tRNA synthetase. Is it clear? So here in order to organize the translation mechanism or the translation process, there is a requirement of two specific strategies, and these include one is adenylation and the other one is tRNA charging. Is it clear? So here by taking the advantage of these two specific strategies, the translation mechanism or the translation process, it commences both in case of prokaryotes and even in case of eukaryotes. So let me arrange the things here. So here the two specific strategies which are responsible for organizing the translation mechanism, and here these include adenylation and the other one is tRNA charging. Is it clear? So let me write it here. Adenylation and the other one is tRNA charging. tRNA charging. So here what is adenylation? Here each and every amino acid which is very specific to one particular codon in case of the mRNA, it has to interact with ATP. So here once when the amino acid, when it interacts with ATP, amino acid, it has become adenylated. Is it clear? So let me write it here. Amino acid, when it interacts with ATP, amino acid, it has become adenylated. Adenylated amino acid. Now here the adenylated amino acid, it successfully recruits to one particular tRNA by taking the advantage of amino acid tRNA synthetase. Is it clear? As I have already mentioned you, in order to organize the translation mechanism, there is a requirement of a specific raw material which is nothing but what? Amino acid tRNA synthetase. So here what is the role of amino acid tRNA synthetase? Here amino acid tRNA synthetase, it is the specific enzyme which is responsible for the successful recruitment of adenylated amino acid to the concerned tRNA. Is it clear? So let me show it here. Adenylated amino acid. Adenylated amino acid. So here the adenylated amino acid, it successfully recruits to the tRNA by taking the advantage of a specific enzyme which is nothing but what? Amino acid tRNA synthetase. So here once when the adenylated amino acid, once when it successfully recruits to the tRNA by taking the advantage of amino acid tRNA synthetase. Now here the specific tRNA, it has become charged. Is it clear? Once when the adenylated amino acid, once when it successfully recruits to the tRNA by taking the advantage of amino acid tRNA synthetase, now here the specific tRNA, it has become charged tRNA. Only charged tRNA, they are capable to participate in case of the translation mechanism. Uncharged tRNAs, they are unable to participate in case of the translation mechanism. Is it clear? And here it is termed as what? tRNA charging. So let me write it here. Adenylated amino acid. Adenylated amino acid recruits to one concerned tRNA by taking the advantage of amino acid tRNA synthetase. Amino acid tRNA synthetase. And here the particular mechanism, here it is termed as what? tRNA charging. tRNA charging. So here only the charged tRNAs, they are capable to participate in case of the translation mechanism. Uncharged tRNAs, they are unable to participate in case of the translation mechanism. Is it clear? So here at the very beginning of the translation mechanism or the translation process, what exactly happens is, the ribosomal subunit, the small subunit of the ribosome, which is nothing but the 30th subunit, it successfully recruits to the mRNA. Is it clear? So here likewise the replication and as well as the transcription, even the translation mechanism, it can be well explained or it can be well studied under three specific phases, and these include initiation phase, elongation phase, and the other one is termination phase. So here during the initiation phase of the translation, the small subunit of the ribosome, it successfully recruits to the mRNA. And here after the successful recruitment of the small subunit of the ribosome to the mRNA, so here the small subunit of the ribosome, it successfully proceeds in 5' to 3' polarity until it recognizes the start codon or the initiation codon. Is it clear? So let me arrange the things here. And if we look at the mechanism of translation, so here the mechanism of translation, likewise the replication and as well as transcription, it can be well studied under three specific phases like initiation phase, elongation phase, and termination phase. So here during the initiation phase of the translation, what exactly happens is the small subunit of the prokaryotic ribosome, it successfully recruits to the mRNA, and here the small subunit of the prokaryotic ribosome, it proceeds in 5' to 3' polarity until it recognizes the start codon or the initiation codon, which is nothing but what? AUG. Is it clear? So let me show it here. So here it is nothing but what? mRNA, right? So here to the concerned mRNA, the small subunit of the ribosome, it successfully recruits here. And here basically the small subunit of the ribosome, it has three specific sites, and these include A site, P site, and E site. So let me show it here. A site, P site, and E site. So here, once when the small subunit of the ribosome, once when it recruits to the mRNA, the small subunit of the ribosome, it proceeds in 5' to 3' polarity until it recognizes the start codon or the initiation codon, which is nothing but what? AUG. So here the start codon or the initiation codon, which is nothing but AUG, it is successfully displaced in the P site of the ribosome. Is it clear? So here it is nothing but what? AUG, right? So here the start codon or the initiation codon, it is successfully displaced at the P site of the small subunit of the ribosome. Is it clear? So let me arrange the things here. Small subunit of the ribosome, it proceeds in 5' to 3' polarity. 5' to 3' polarity. And here these are nothing but A site, P site, and it is nothing but what? E site. So here once when the start codon or the initiation codon is AUG, so here in order to recognize the start codon or the initiation codon, the specific initiated tRNA with its encoding amino acid, it comes into the picture. So here once when the start codon is AUG, the initiating amino acid, it is not neither methionine nor it is valine, but it is a modified form of methionine, which is nothing but what? Formulated methionine in case of prokaryotes. Is it clear? So let me show it here. Initiated tRNA with its encoding amino acid, it comes into the picture. So here it is nothing but what? Formulated methionine. So here once when the initiated tRNA with its encoding amino acid, once when it recognizes the start codon or the initiation codon, in the very next step, the large subunit of the ribosome, it comes into the picture and it remains in association with the small subunit. Is it clear? So let me show it here. So here it is nothing but what? The large subunit of the prokaryotic ribosome. So here once when the large subunit of the prokaryotic ribosome, once when it remains in association with the small subunit of the prokaryotic ribosome, with this, the initiation phase of the translation mechanism in case of prokaryotes, it has successfully organized. Is it clear? Association of the large subunit of the prokaryotic ribosome with the small subunit of the ribosome, it indirectly suggests or it indirectly says that the initiation phase of the translation mechanism in case of prokaryotes, it has successfully organized. Is it clear? So here in the very next step, elongation phase of the translation mechanism, it comes into the picture. So here in what way? During the initiation phase of the translation, the initiated tRNA with its encoding amino acid, it is capable to recognize the start codon known as AUG. In the same way, during the elongation phase, the amino acid tRNA with its encoding amino acid, it is successfully displaced at the A site of the ribosome. Is it clear? So here it is nothing but what? Amino acid tRNA with its encoding amino acid. So here once when the amino acid tRNA with its encoding amino acid, once when it is placed at the A site of the ribosome, now here in between the initiated tRNA encoding amino acid and the amino acid tRNA encoding amino acid, peptide bond formation it takes place. Is it clear? And here in order to organize the peptide bond formation in between the initiated tRNA encoding amino acid and the amino acid tRNA encoding amino acid, a specific RNA it comes into the picture, and here it is nothing but what? 23S rRNA. So here the 23S rRNA in case of prokaryotes, it plays an important role in organizing the peptide bond in between the initiated tRNA encoding amino acid and as well as the amino acid tRNA encoding amino acid. Is it clear? So let me write it here. 23S rRNA in case of prokaryotes responsible for peptide bond formation. Peptide bond formation. Is it clear? And if you look at the chemical reaction which occurs in between the initiated tRNA encoding amino acid and as well as amino acid tRNA encoding amino acid, easily you can understand how the peptide bond formation it takes place. So just look at here. So here it is nothing but what? The initiated tRNA encoding amino acid which is nothing but formulated methionine. Formulated methionine, right? And here it is nothing but the amino acid tRNA encoding amino acid. Amino acid tRNA encoding amino acid. So if we look at here, the specific oxygen atom which is a part of the carboxyl group, it undergoes nucleophilic attack or nucleophilic cleavage. So here once when the oxygen atom which is a part of the carboxyl group of one particular amino acid, once when it undergoes nucleophilic attack or nucleophilic cleavage, now here the oxygen atom it is removed and it is replaced by the nitrogen atom which is a part of the amino group of another amino acid. Is it clear? So let me show it here. So just look at here. It is nothing but what? 23S rRNA which is having the peptidyl transferase activity in case of prokaryotes. Peptidyl transferase activity. Peptidyl transferase activity, right? So just look at here. The oxygen atom it is removed and it is replaced by the nitrogen atom which is a part of the amino group of another amino acid. So just look at here. Is it clear? So here after the successful formation of the peptide bond formation in between the initiated tRNA encoding amino acid and as well as the amino acid tRNA encoding amino acid, now here the initiated tRNA which is under uncharted conformation, it successfully translocates from the P site to the E site. P site to the E site, right? So here it is nothing but what? Initiated tRNA which is under uncharted conformation. Uncharted conformation. So here as I have already mentioned you, only the specific tRNAs which are under charged conformations, they are capable to participate in case of the translation mechanism. Uncharged tRNAs, they are unable to participate in case of the translation mechanism. So the reason why, once when the initiated tRNA, once when it participates in the peptide bond formation, now here the specific initiated tRNA, which is under uncharged conformation, it successfully translocates from the P site to the E site. So here, once when the initiated tRNA, once when it successfully translocates from the P site to the E site, now the P site of the ribosome is free. Now here the P site of the ribosome, it is occupied by taking the advantage of amino acid tRNA. Is it clear? Amino acid tRNA, it successfully translocates from the A site to the P site. So here, once when the amino acid tRNA, once when it successfully translocates from the A site to the E site, now the A site of the ribosome is free. Now again, the A site of the ribosome, it is occupied by taking the advantage of one more other amino acid tRNA with its encoding amino acid. So here, after the successful recruitment of amino acid tRNA with its encoding amino acid at the A site of the ribosome, then after, again the peptide bond formation occurs in between the two amino acids by taking the advantage of 23S rRNA, which is having the peptidyl transferase activity. Is it clear? So let me show it here. So here it is nothing but what? Polymerization of amino acids, which lead to the formation of polypeptide chain or protein. So here these series of sequence of events, they proceed until the small subunit of the ribosome, it is about to reach the stop codons or the termination codons. So here we need to understand that the small subunit of the ribosome, it is capable to recognize only the sense codons which are present in case of the mRNA. And here the small subunit of the ribosome, it is unable to recognize the stop codons or the termination codons. And even no specific tRNA, it is capable to recognize the stop codons or the termination codons like UAG, UGA and UAA. So here, in order to recognize the stop codons or the termination codons, the small subunit of the ribosome or the specific translation mechanism, it takes the advantage of a specific factor which is nothing but what? Release factor. Is it clear? So let me write it here. Release factor. Release factors are used during the termination phase. Termination phase of translation. So here, after the successful formation of the peptide bond in between the two amino acids, the elongation phase of the translation mechanism, it has successfully organized here. So here, within no time, the termination phase of the translation, it comes into the picture. And here, during the termination phase of the translation, the small subunit of the ribosome, it is capable to recognize the stop codons or the termination codons like UAG, UGA and UAA. So here, in order to recognize the stop codons or the termination codons, the small subunit of the ribosome, it takes the advantage of specific factors known as release factors, because the small subunit of the ribosome, it is capable to encode only the sense codons, and even the specific tRNAs, they are capable to encode only the sense codons. No tRNA is capable to recognize the stop codons or the termination codons. In order to recognize the stop codons or the termination codons, the specific translation mechanism, it takes the advantage of a specific factor known as release factor. So here, by taking the advantage of specific release factors, here the small subunit of the ribosome, it is capable to recognize the stop codons or the termination codons. So here, once when the small subunit of the ribosome, once when it is capable to recognize the stop codons or the termination codons, under such condition, the large subunit of the ribosome, which is in association with the small subunit, it undergoes dissociation. Is it clear? As I have already mentioned you, both the ribosomal subunits in case of prokaryotes and even in case of eukaryotes, they remain in associated form only during the active conformation, and during the inactive conformation, the large subunit of the ribosome, which is in association with the small subunit, it undergoes dissociation. And even the polypeptide chain, which has successfully synthesized during the translation mechanism, it also successfully dissociates from the ribosomal machinery after the successful organization of the termination phase of the translation. Is it clear? So let me show it here. So let me arrange the things here. So here these are nothing but what? The ribosomal subunits, right? Ribosomal subunits. So here it is nothing but the large subunit, and here it is nothing but what? The small subunit. So here, after the successful recognition of the stop codons or the termination codons, here both the ribosomal subunits, they undergo dissociation, and even the polypeptide chain or protein which has successfully synthesized during the translation mechanism, it also successfully dissociates from the ribosomal machinery. So here with this, the translation mechanism or the translation process, it has successfully organized in case of prokaryotes. Is it clear? So here this is regarding the translation mechanism or the translation process which is successfully organized in case of prokaryotes. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. Thank you.